Today we'll talk about gene therapy and gene editing. And let me start with a simple yet somewhat loaded question. Why do we get sick? What makes us sick? Viruses, bacteria, larger but still small. Our genes, we go back to that. So viruses, bacteria, parasites, those are actually way worse. Like we immediately gravitate to viruses and bacteria for, I suppose, all of the right reasons, but parasites are real human killers. But then, yes, sometimes we get sick because of our own genes. There is something that is not working properly with our genes and we get sick. And it can go anywhere from having a certain predisposition to disease, depending on certain environmental inputs, all the way to, being sick because of these very penetrant genetic disorders that are sometimes explained by mutations in a single gene, right? So forever, we've treated those conditions symptomatically. We try and fix whatever is wrong in those conditions or genetic diseases by attacking the symptoms and trying to counteract them. But since the advent of molecular genetics and our ability to engineer genes, obviously a very obvious idea came about, and that is, why don't we try and fix those genetic problems within the patient? And that is what we call gene therapy, is treating a disease by introducing into the patient or correcting in the patient any genetic abnormalities that there can be. And there is two very broad approaches to gene therapy. We call them ex vivo, when the affixing of the genetic mutation happens outside of the patient, or in vivo, when we're fixing those genetic problems inside of the patient. So let's talk about the in vivo approaches first. These are the simplest conceptually. The idea is you can apply these type of approaches whenever there is a genetic function that is missing in a person. So this is very common for monogenic disorders that are caused by mutations, typically recessive mutations that basically make a function not be present in the individual. So think those diseases that have to do with lacking a important metabolic enzyme or some sort of protein that is important for some cellular function, et cetera, et cetera. So the approach conceptually is rather straightforward. The patient is lacking a genetic function. What we do with in vivo gene therapy is we put it back in. And the method of choice for this type of approach has been the use of adenoviruses. Adenoviruses are, in nature, DNA viruses that can infect our cells and make us sick. But in the lab, we've hacked these adenoviruses and engineered them such that we can use them to introduce genes that are good for us, or genes that we would like to introduce into the cells of a patient, right? So the cartoon here is showing how you have a patient suffering a condition, and you have the gene that you would like to introduce into that patient. What you do is you package that gene into a modified adenovirus. That adenovirus will infect the cells in the organism, bring with it the DNA cargo all the way into the nucleus. Adenoviruses have a way to inject DNA into the nuclei directly by latching themselves to nuclear pores. And then that DNA will go into the nucleus, and as it is stressed here, without integrating into the genome, it can start being expressed. It can start being transcribed, processed into mature mRNA molecules, and then start producing whatever protein was missing. Why do we emphasize that they do not integrate into the genome? Because that's actually good news for safety. If the DNA that you're putting into a patient's cells does not integrate into the patient's host or into the patient's genome, so the patient's own genome, there is no risk of accidental mutations. There is no risk of interrupting any of the genetic sequences of the patient. There is no risk of any of these interruption of, for instance, tumor suppressor genes that might lead to the formation of a cancer or something like that. All of these things are problems that arise with other technologies that we'll discuss today. But that's not a problem with adenoviruses. They do not integrate into the genome. So all you're doing is you're putting some DNA into nuclei for a while, because that DNA is kind of doomed to be destroyed at some point. The host cell will eventually, or the infected cell will eventually recognize that it has been infected. So it will eventually destroy that DNA. But until that happens, that DNA can be expressed and it can provide that missing function, that missing genetic function for the patient. Here's a list, relatively updated list of gene or cell therapy approaches that have been approved by the FDA. And there's a list right there, and the ones that I highlight here are examples of conditions treated with adenoviruses nowadays. So here we have an adenovirus that is designed to produce an immunity stimulant in cancer cells. The idea here is that cancer cells often evade immune attack by the patient's immune system by sort of flying under the radar, if you will. And there are some adenoviral-based gene therapies that what they do is they make those cancer cells more visible to the patient's immune system. So now that the patient's immune system can attack them. There is an example of an adenovirus designed to produce a retinal pigment protein in patients that normally have reduced vision because they're lacking this protein. So now you're simply making retinal cells begin producing this protein and vision improves. And last, an adenovirus designed to produce this somatic motor neuron protein to prevent these motor neurons from dying and causing a neurodegenerative condition. So a handful of examples of therapies that have been approved and are in use in the clinic nowadays. Okay? Now, there are some disadvantages to these type of approaches, some challenges. One is that you have to constantly re-inject these engineered adenoviruses because, like I explained just now, the infected cell will produce the therapeutic protein for a while, but then eventually will recognize that foreign DNA as foreign and it will destroy it. So there is a need for constant re-injection. And perhaps more importantly, one of the biggest limitations of in vivo adenoviral gene therapies is that sooner or later, the patient's immune system will recognize these modified adenoviruses and mount an immune reaction against them. It's almost like you're injecting the patient with a vaccine every time. So at some point, the patient's immune system may learn to recognize these adenoviruses and attack them pretty violently. So there is, from the patient's perspective, there is a fever, some chills and muscle ache, not much more than that. But from the therapy perspective, you've lost your therapy because now you're injecting these adenoviruses that go nowhere. Right? So that forces the doctors to find either different versions of the similar adenoviruses, try and evade that mounted immune reaction, or go for some other alternative therapy altogether. Okay? On the other hand, we have these ex vivo therapies, and what this cartoon is supposed to show here is the approach goes as follows. You have a patient with a given condition. Again, typically these are conditions caused by the lack of a given gene, so you're trying to reintroduce a gene into this patient. So what you do is you take out some of those cells from the patient, you modify them genetically outside of the person in a cell culture dish, if you will, or a test tube. And then once you have obtained genetically modified cells from the patient, you amplify them, once again in vitro, outside of the of the body, and then you re-inject them or reintroduce them as soon as you have enough of them. And now these genetically modified cells will do whatever they were not doing before. Okay? For this type of approach, things are changing, of course, so we're going to discuss what has been approved and is being used nowadays. This is changing as we speak with the advent of gene editing technologies. So what we'll discuss in the next few slides is what is being done and what is approved, and we can go to the doctors and for some conditions, obviously, get these therapies. For approved therapies, what is most commonly used are retroviruses, and retroviruses do integrate themselves into the genome. So the way that a retrovirus normally lives its life is more or less as follows. The virus will find a target cell, will fuse itself with its cell membrane, and it will dump inside of the cell 2 or 3 key components. First of all, it will introduce its RNA-based genome. Right? So all of its genetic information in the form of an RNA molecule will be dumped into the infected target cell. I can't remember what is what with these little cartoons. I think the little blue dots here are supposed to represent enzymes that we've discussed before, that we heard about before. These famous reverse transcriptases or retrotranscriptases. These are enzymes that can read an RNA template molecule and synthesize a DNA molecule complementary to it. Right? So if we zoom in real quick here, there is this process of reverse transcription by which an RNA molecule is used as a template and a cDNA or copy DNA molecule is made by these retrotranscriptases. And then those cDNA molecules are in turn used by generally the same retrotranscriptases to make the second DNA strand. So that's how we go from a single-stranded RNA molecule to a double-stranded cDNA. And that double-stranded cDNA is what integrates itself into the host cell's genome. And it uses a mechanism that is not much different from what we discussed for transposons on Tuesday. It will use very long repeated sequences that are flanking, that are on both ends of these long double-stranded cDNA molecules now, to sort of induce a recombination event that will place them inside of the host cell's genome. I emphasize the need for those long repeats, or as we'll see in a few seconds, they're called long tandem repeats, because it's not much different from what the Cre-Lox system does with those LOXP sequences. Okay? This is the natural life of certain retroviruses. And possibly one of the most famous retroviruses in humans is HIV. Right? This is what the HIV virus does. It will integrate itself into the genome of specifically T4 cells, of immune cells, and hence that's how they cause eventually immune deficiency, because they specifically kill immune cells. Now, a retrovirus can be integrated into the host cell's genome for a very long time in what we call a latent phase. Right? So the sequence is sitting there, but it's not really doing much to the cell. At some point, and via mechanisms that in many cases are not fully understood, the retroviral DNA integrated into the cell's genome decides to activate, and it will start producing essentially 2 things. Many copies of the original RNA genome that made up that retrovirus that infected the cell. So there will be, through transcription, a lot of copies of this RNA genome made. And some of those RNA molecules will be used to translate a whole bunch of the proteins that the virus would need to make new viral particles. So now you have all of these proteins that are needed to make a new viral particle and the RNA molecules that would give those viral particles their genetic information, they get assembled and packaged inside of the cell, and then they get extruded through this lytic process by which the cell is producing so many of these viral particles that it essentially dies in the process. Right? So when the retrovirus decides to activate and kickstart this process, the consumption of resources and energy within those cells is such that the cell dies. Okay? So that's what these retroviruses do naturally. Why did do that? And like I said before, alluded to before, they do that because they all share some generic structure. And that generic structure includes these LTR sequences, or long tandem repeats. What that means is there is these sequences that are repeated in tandem several times, so long tandem repeats. And they flank sequences that are important for this cycle of integration into the genome and later the lysogenic or lytic phase, right? So it's the proteins that make the viral capsid. Those are encoded by GAG genes. There is the polymerases that will make or encode for those enzymes that are important for reverse transcription and genome integration. And then there is the ENV genes that make the glycoproteins and the capsid proteins that are important for encapsulating new viral particles. So all of those things exist naturally in retroviruses and are important for a retrovirus to not only infect a cell and integrate itself into the genome, but also produce new molecules. What scientists did in the lab is, once again, and this is not much different from what happened with that TI plasmid from the Agrobacterium tumefaciens that we discussed on Tuesday, separate some of the biological functions that these retroviruses normally carry out from the engineered functions that we would like them to carry out for us. Right? So the way this looks is scientists placed a whole bunch of therapeutic sequences between these LTR or long tandem repeat sequences, right? Not much different from what we do in plant biotechnology when we introduce our sequences of interest between the TL and TR sequences, right? So once again, it's a recurring theme. Anytime that you want to integrate something into a host genome, you put it in between these, in this case, the LTRs. In the case of Agrobacterium, it was the TLTR. In the case of Cre-LOX recombination, it would be between the LOXP sites, right? So you engineer the sequences you want to place into a target cell between the LTRs. Then you provide all the other functions that these retroviruses need separately, and in a way that those things don't make it into the target cell. So it's almost like you let the cells prepare these viral particles with everything they need, but then the only thing that they inject is the sequences that can integrate themselves into the genome without letting them replicate again themselves. Right? So we detach the replicative ability of these retroviruses from the ability, their ability to introduce sequences into the genome. And the way this is done is either by using two different plasmids, or in some cases even three plasmids. So here on the left side, what is shown is that you can introduce into cells, and there's many cells that you can use for this, but into cells you introduce one plasmid with a strong promoter and the sequence for the ENV genes that are important for making the capsids, and then another plasmid with a strong promoter and then the GAG and the POL genes. Again, what these will do is just make the proteins and the enzymes that your sequence of interest will need to infect the cell and integrate itself into the genome. But those sequences that infect themselves, that make it into the genome, that are contained between the two LTR sequences, don't include any of these enzymes that are critical for making new viral particles, right? So you've killed the ability of these retroviruses to replicate themselves in the target cells. Because this technology works so well, eventually some companies created cell lines that have the GAG, POL, and M genes in their own genome. So there is these special cell lines that you can order from companies that will package retroviruses for you. And when you package a retrovirus, this cartoon here shows how there is a transfection, or if you're using one of those cells with the M, GAG, and POL genes in them, you don't need to worry about transfecting a couple of those plasmids. But bottom line is you introduce a vector with LTR sequences flanking the sequences that you would like to integrate into the patient's genome. You let these cells produce a whole bunch, and we're talking millions of viral particles containing your therapeutic sequence of interest between LTR sequences. And then you take those viral particles, and in the process of transduction, as the technical name given to the infection of cells by these type of retroviruses, you let your DNA of interest with your therapeutic sequence of interest not only go into the cell, but make it all the way into integration. And once integrated in the genome, and provided that you've given these sequences, therapeutic sequences of interest, the right promoters, they will start producing mRNAs for your recombinant therapeutic protein. They will not produce the reverse transcriptase or the capsid proteins or the glycoproteins or any of those things that a normal retrovirus would normally need to package new viral molecules. Huh? Did that make sense? So in a lab, and these days in a company, in a lab within a company, you would produce these retroviral particles with a given therapeutic sequence of interest. Then you purify those viral particles, and then you inject them into a patient and let those retroviruses go and infect the target cells. What target cells will be infected depends on a number of factors. Some of them can be controlled by how you encapsulate these viruses, because depending on their encapsulation, you may be able to target specific cell types. So if you were using a capsule from, say, a virus like HIV, you would be targeting immune cells of the T4 kind. If you coat these retroviruses with other proteins, you may be targeting other target cells. Okay? Cool. So, just like, well, so I don't have a slide for that, but one of the advantages of this type of gene therapy is that you don't have to worry about reapplying the therapy over and over. Once you've generated enough of these genetically modified cells that you can reintroduce into the patient, those cells will most likely propagate themselves and sort of perpetuate themselves through replication. So you don't have to worry about re-injecting the patient over and over again, and therefore there is no risk of the patient mounting some sort of immune reaction. So that's an advantage. The challenges with retroviruses, the disadvantage is that that integration process is typically random. So there is always the risk that wherever that engineered sequence will integrate itself into the target cell's genome, it may break genes that are important for keeping tumors in check, and next thing you know, you created a tumor. Ex vivo has that increased potential for inducing mutations that could be tumorigenic. Now, of course, companies have ways to the best of their abilities, try and prevent these things, or at the very least, have some sort of heads up that this may happen. How? Well, don't forget that when you introduce these sequences into the genome with a retrovirus, you can always use selection pressure, so you can always integrate into the cell's genome a selection marker as well, resistant to some sort of antibiotic, such that you kill all the cells that do not integrate the engineered sequence, and you can maintain only the ones that did. And among those, you can let them amplify several times, and then with a relatively small sample and modern molecular genetics techniques, you can fairly accurately ask the question, where did this thing integrate itself? Right? All you need to do is do a little fancy sequencing around the site of integration, and you will find out quickly where this thing integrated itself. So companies, for the most part, have ways to know that a given therapeutic transgene of interest integrated in a place that is safe, that didn't break any tumor suppressor genes or is not going to cause any kind of genetic instability. Now, all of that is easy to say outside of the patient, right? The context in a lab is different from the context inside of the patient. So there's always an element of risk with these therapies that once you reintroduce these cells into the patient, there may be some posterior and what's the word that I'm looking for? Unpredictable later events that could lead to the formation of tumors. And in fact, that has been, things are changing, but that has been one of the things that has kept gene therapy as an approach to medicine, has kept it sort of in check for decades. We've conceptually known about these ideas of introducing therapeutic sequences in patients for decades. Late 70s, early 80s, there were already people thinking about these things because the technologies were becoming available for genetic engineering. It's just that there were, in the early days of gene therapy, there were some really nasty examples of patients in clinical trials for these therapies that died because of the tumors produced by these recombinant cells that were introduced back into them. So for a while, gene therapy had to take a back seat and no one dared to go there. With improvements to these technologies that allow us to know in advance where these transgenes have integrated themselves and with a high degree of certainty, there's still that element of the unknown that, well, who knows, within the biological context, this place that seemed completely inert in a culture dish in the lab, in the patient, is super important, and now we're causing some trouble. But the risk has diminished quite a bit. And then the other common challenge with ex vivo gene therapies is you spend a while getting the cells from the patient, amplifying them in vitro in the lab, then you genetically modify them. Not all of them will, obviously. These technologies are notoriously inefficient, so only a handful of the cells in your large collection will be genetically modified. Then you go through the screening process for selecting the ones that, to the best of your knowledge, are not going to cause any trouble later on. Then you amplify those, and then you inject them back in the patient. And for reasons that no one really knows, they don't engraft well, meaning that those cells will either be recognized as foreign cells and be attacked by the immune system, or they just don't proliferate in the organism. They were doing fine in vitro, then you put them back in the body, and for some reason they stop dividing. So that's called poor engraftment of recombinant cells, and that happens. And that is one of the more common limitations of these type of approaches nowadays. And if you want to do it again, you can, but you have to go through the whole process again. And you might be able to do it again anywhere between tens and hundreds of thousands of dollars later, because these things are still expensive. So with that said, there are some of these ex vivo retroviral gene therapies that have been approved for use in patients, and those are some examples there. And one approach that we don't have all of the examples there, because many of them are still undergoing clinical trials and whatnot, you may have heard about, and that is the approach of cell-based immunotherapies against cancers, right? These by a show of hands, has anyone heard of these before? CAR T cells? CAR T cells. Kind of rings a bell, CAR T cells. It's the newest thing. It was actually developed in UCLA, so down the road somewhere. And the idea is you take immune cells from the patient and then you reprogram them genetically, you engineer them genetically to specifically recognize cancer cells of certain types, right? So you take immune cells from the patient, you teach them genetically how to recognize cancer cells, and then you put them back in the patient. And CAR T stands for cell antigen recognition T cells or something like that. Don't call me on that, but it's the new thing. And there are many companies that are now starting to deploy this technology against different cancer types, because all a company really needs to do today is demonstrate that they've homed in on a protein that is expressed only by a certain cancer cell type, and that the modified CAR T cells can get to them. Other than that, everything that goes through, everything that is related to the genetic engineering of these cells outside of the patient, all of those wrinkles have been ironed, and all the T's have been crossed and I's dotted, such that the FDA and the NIH know now that the early steps are all taken care of and can be done safely. All that companies need to demonstrate to the FDA today is that they homed in on a gene that can lead these CAR T cells to the tumor cells and kill them. Yeah? So an example, and I know, like if you zoom in, you'll see down here, there is Kite Pharma down there. Again, like somewhere on the other side of the hills. Kite was one of the first companies that developed these CAR T technologies, and they've been producing therapy since. Everything that we've discussed so far is, in a way, patchwork, right? So there is a gene that is missing or broken in the patient, or you can think of a gene that if you were to introduce in the patient, it would do something good, like kill cancer cells. So what you're doing is you're providing these genetic sequences that are missing separately, right? But let's not forget that the problem originated for some of these monogenic diseases, right, originated from the patient's own gene being broken, right? Being mutant, being missing. But let's say that there is a mutation. So if you really wanted to fix the problem from scratch, instead of providing these genetic sequences separately with adenoviruses or retroviruses that integrate themselves in the patient's genome elsewhere, wouldn't it be cool if we could go in and fix that problem to begin with, right? And that is what nowadays we refer to as gene editing. You're going into the patient's genome and editing specific sequences that underlie some sort of genetic condition. Okay? So let's imagine that there is a given gene, and the cartoon here shows that little red asterisk to indicate, for instance, a point mutation. Right? And I know I make it sound like it's just some silly example, but many monogenic diseases are caused by known point mutations that make those genes non-functional. Right? So my question to you guys now is, how do cells fix sequences that they perceive to be abnormal? What mechanisms do cells use to say, there's something wrong here, I need to fix this sequence? Correct. What kind of recombination? It has a very specific name. You're so close. Recombination is the second word. Starts with ho, ends with homologous, right? Homologous recombination, that's right. So there are instances in which cells can tell that there is something wrong with the sequence, and they can carry out homologous recombination to replace the wrong sequence, quote unquote, with the right one. And they do that when they have access to that, quote unquote, right sequence. And they make those determinations based on nearby homology or surrounding homology, right? Now, many other times in this class, we've discussed how if things look okay from a biochemical perspective, from a biochemical point of view, if there's nothing chemically wrong with any region in DNA polymerases, DNA or RNA polymerases, they have no way to tell that a sequence is quote unquote wrong, right? How many times did we discuss that you can actually introduce changes or mutations of interest because you can later fool the cellular machinery into thinking that that particular nucleotide was there all along, right? Like we've said this before in class. So homologous recombination won't really kick in until there is something more seriously wrong with that region of DNA, right? So theoretically, you could introduce the therapeutic sequence that you would like to fix the problem with and hope for homologous recombination to replace the mutant variant or the broken sequence. But homologous recombination on its own, it's an exceedingly rare event. It occurs in less than 0.1% in cell culture. And we discussed this before, how whenever there is, let's say that you introduce this therapeutic sequence as a vector, right? And you hope for homologous recombination, that's going to be a very rare event. It will happen when the cell recognizes the circular DNA molecule and it goes, oh, well, this must have broken off from the genome. Let me put it back. Where should I put it back? Oh, let me see if I can find a homology region so that I can induce homology recombination. But on its own, it's an exceedingly rare event. It can happen much more frequently when DNA breaks, when DNA is broken. Right? Because now those broken DNA molecules will trigger a series of alarm signals within the nucleus and by extension the cell that will accelerate this process of homologous recombination. And that's where conceptually gene editing comes in. The idea here is if we can target DNA breaks to where the problem is originally, and we can induce a double strand break where, let's say, if not exactly at where this mutation is, at the very least nearby, and we offer a therapeutic sequence with homology regions around it, then we can hope for a much more frequent homologous recombination event that will replace the broken sequence with our therapeutic sequence of choice. Now the question, of course, is how can we precisely cut DNA at a specific location? How can you cut DNA to start with? Like, how would you go around cutting DNA? What? Yeah. Restriction enzymes, right? That's what you've learned so far, restriction enzymes. What's the problem with restriction enzymes, though? They're not that specific, are they? Right? We had some calculations basically to say that the probability of a restriction enzyme finding its target sequence in a genome can be calculated as 1 divided by 4 to the power of n being the number of nucleotides in its recognized target sequence. So for a restriction enzyme that recognizes 6 nucleotides, that's sure a small number, but it's a small probability. But when you think how big the genome is, those restriction enzymes will cut, say, the human genome thousands of times, right? So you need something that is way more specific than a restriction enzyme. Can you guys think what that might be? It's famous, it's in the news every now and then now. CRISPR, yes. So show hands again, who heard of CRISPR? Not that you could explain to me what it is. Yeah, most of us heard about CRISPR. It's the thing these days, right? So that's right, CRISPR. So what is CRISPR? Sometimes referred to as CRISPR/Cas9 for reasons that we'll understand in a sec. So CRISPR, let's start with this. As usual, it's something that was found in the wild. CRISPR is a defense system that bacteria evolved to defend themselves against recurrent infections by the same bacteriophages over and over again. So we're not going to go into the details, but the CRISPR system was invented by bacteria, and they use that system as, if you will, an adaptive immune system. What I mean by that is bacteria using CRISPR learn about prior infections in their history, and they are better prepared to repel those infections later on. As usual, people in labs learned about this system and hacked it. Why is CRISPR so cool? Because it, unlike restriction enzymes that will go and cut DNA in a specific but very frequent manner, meaning they will recognize certain sequences in DNA, but they recognize those sequences very often, CRISPR is based on much longer complementarity to the target sequence. The enzyme that does the cutting of the invading DNA in bacteria is called Cas9, and that enzyme uses a molecule that we call a single-stranded guide RNA, or sgRNA, to find its target sequence. And that sgRNA is not 4 or 5 or 6 nucleotides long. It's closer to what we saw for microRNAs and siRNAs. So it's somewhere around 20 nucleotides in length. So if you do the same math, right, and now you ask yourself, how often will I find the target sequence for the sgRNA that is loaded onto a Cas9 enzyme in a genome? You can calculate that as 1 over 4 to the power of 20. That number now is astronomically low. What that translates to is the chances that a Cas9 loaded with a given sgRNA will cut in more than one place in the genome are almost zero. These Cas9s with a given sgRNA loaded onto them are exquisitely specific about where they will cut DNA. They will cut only if they find that complementarity to their sgRNA for near 20 nucleotides. So that won't happen by chance. That will be exceedingly specific. So the way we use CRISPR-Cas9 in genetic engineering or genetic editing is we now know where we would like to introduce a double-strand break in DNA. All we need to do is design an sgRNA that surrounds that cut site that we'd like to target. So we design an sgRNA knowing the sequence before and after that cut site. Then in vitro or in cells, we assemble the Cas9 with that sgRNA, and then we expose the target cells to that complex. And the Cas9 sgRNA will patrol the nucleus and look for its target sequence in the host cell's DNA until it finds it, and it will introduce those double-strand breaks. At that point, that double-strand break will happen not several or many or thousands of times across the genome. It will happen only in one position. And as long as, once again, you offer some sort of therapeutic sequence flanked by homology sequences on both sides, you can count these cells now, having detected that double-strand DNA break, engaging in homologous recombination to try and fix that problem and replacing the old mutated sequence with the new therapeutic one. So what I just discussed is, in a way, a progression of the genomic integration of retroviral sequences, except that it's not random. It's exquisitely specific. So if you can take your cells from the patient and then use CRISPR-Cas9 to direct the double-strand break to a very specific location of the genome and homologous recombination to fix that cut, now you have genetically modified cells from your patient that you can amplify and reintroduce in the patient. Are we doing that yet? Well, not quite yet, because there are still some safety hurdles to go through when it comes to CRISPR-Cas9. We haven't really crossed all those T's and dotted all those I's when it comes to safety. Are we do we know for sure that it's cutting only one place? Could it have some sort of non-specificity when it comes to the sgRNA? Maybe it doesn't base pair throughout the entirety of those 20 nucleotides, but what happens if it base pairs in 17 out of those 20 positions or 16? Could there be a cut in that case, and so forth, right? But we're getting there, and slowly but surely the technology is improving such that it's becoming safe enough that at some point, instead of using retroviruses for ex vivo gene therapy, we'll be using CRISPR editing. Okay? So, what's the plan? Well, let's say sooner, what we can expect is companies taking cells from the patient and, as I said before, deciding on what sequence they would like to target for CRISPR and deciding what sequence they would like to introduce via homologous recombination, and then mix everything together, the target cells, the Cas9, the sgRNA, and the therapeutic sequence for homologous recombination in a dish, recombine or edit those cells in vitro, let them proliferate in vitro, and then reintroduce them in the patient. So this is happening as we speak. There are other people looking into, let's introduce the machinery for CRISPR into the cells using, for instance, adenoviruses. So why not try an adenoviral approach by which what you do is you load up those adenoviruses with the sequences that you need for making Cas9, making the sgRNA, and making the homologous recombination therapeutic sequence, or the therapeutic sequence to be recombined into the genome through homologous recombination. And then you put everything into an adenovirus, you let the adenovirus infect the cells, and when the cells are infected, they will start expressing the 3 components needed for CRISPR, and they will do it. Right? So that's another approach that is being actively researched. So you don't have to mess around with cells in vitro, which always introduces an element of risk, because you've taken cells out of their context and messed with them outside in a plastic dish. If this adenoviral approach were to work, then you let the CRISPR machinery do its thing inside of the organism. Okay? But now let's talk about something that is a little more fun, in the sense that it is a little more captivating of our imagination, and it can make for fun conversations at dinner parties. And that is the concept of designer babies. Had you heard of that before? Right? It's very simple. Conceptually, from the moment societies heard we can edit DNA, immediately they gravitated towards thinking about, so now we can edit a baby to be whatever we want. A super musician, a super athlete, a super soldier, a super genius, right? The thinking is, if you can take either a zygote, or in some cases a gamete, and use CRISPR to edit a gene of choice into that zygote or gamete, you've basically introduced a new gene variant into the human genome. And you let those zygotes or gametes fertilize and develop into a baby, and you've designed genetically your baby. Here's the water split. Let's get real about this. So here's where the water split. There are some people thinking and working very actively on this approach for medical reasons. And the idea goes, when there is, for instance, a couple which either parent or both are carriers of certain genetic conditions, and they don't want to risk having children that suffer that condition, what you could, in principle, do is let the zygote develop to the point that you can edit it genetically, fix whatever the problem is, and then let that embryo develop to term, and you fix a genetic problem associated with a disease. So that's real, that is happening, and there is very few people who, I mean, there are obviously some ethical concerns, but there is very few people who get up in arms with the idea of fixing a genetic mutation before you let the embryo develop. The whole story about I can take your zygote and make a super cello player or a super athlete, we're not even near, right? And the reason for that is, what is the gene associated with cello playing? Never heard of celloing one? Doesn't exist. What's the one gene associated with being the super NBA star? Doesn't exist. We're talking hundreds of genes that contribute to those types of abilities. Genes and environments and genetic by environment interactions, right? So this idea that you can, editing one or two or even a handful of genes, make a super athlete, super musician, super genius, science fiction, okay? And as usual, science fiction sometimes sort of drags the real applications of these powerful technologies, like it is conceivable that for certain monogenic diseases, instead of having to worry about those things later on with ex vivo or in vivo gene therapies, if you could fix it at the time of embryo development, early embryo development, why not? And last, we're going to use the last 5 minutes to discuss briefly another very potent application of gene editing, and that is related to the concept of gene drives. Now I need you guys to take off your human medicine hat and put on a sort of an ecological restoration hat, right? There are many ecosystem level problems that could be solved via gene editing. Think, for instance, malaria. So imagine if we could make mosquitoes that resist infection by the malaria parasite and don't transmit it to humans anymore. Think about, I understand that for many of us, global frog mortality doesn't seem like much, but it turns out that it's a serious ecological problem that is revealing something much more serious happening with ecosystems. And now we know that these frogs are dying en masse due to infection by certain fungi. So what if we could make these frogs resist those fungal infections? And you may have heard of pollinators collapse, more specifically collapse in bee populations that have many farmers very, very scared, because if you lose bee pollination, you may lose entire crops and industries. So imagine if we could make bees that resist pesticides and pests that are underlying these collapses in the population. Now the problem is that, let's say we genetically engineer a bee. We're going to use the bees as an example. Let's say that we genetically engineer a bee in the lab such that we introduce into this bee a pest resistance gene. And we use CRISPR, so it's going to be exquisitely specific where this thing goes, safe, nothing else gets touched in the genome. You make your bee, and because these technologies, as we've discussed several times, carry with them a certain degree of inefficiency, chances are that at first, all you'll get is a heterozygote individual. You will have edited only one copy or only one of the homologous chromosomes. So you have your heterozygote B, right? But you can do this a number of times, since you're always using the same sgRNA to target the gene editing. You can probably get more than one heterozygote B in your lab. So all you need to do now is cross those Bs. Not that easy with bees because of the whole queen and drone thing, but it can be done. You cross those bees and then you get a homozygote bee. So now that bee is homozygote for the edited gene of your choice, and you release it in the wild. That bee will obviously, or we can think of it, let's bring it back. I got carried away with the bees. Let's think about mosquitoes and malaria. And here's the reason why. These experiments have been tried with mosquitoes. Taking mosquitoes that have been found to be resistant to malaria through other mechanisms, people will grow them in controlled environments and they will release them into the wild in the tens of thousands. They bring these weird cages with tens of thousands of mosquitoes, and then they release them in the wild. Tens of thousands versus the tens of millions in the wild, chances are that your homozygote, say, mosquito resistant to malaria, will mate with a wild-type mosquito. And then what you'll have is a heterozygote mosquito in the progeny, only half of the progeny, Mendelian ratios, right? So one homozygote for the malaria resistance, one wild-type, half of your progeny will be all of your progeny will be heterozygote. And when that heterozygote mosquito mates again in the wild, it will most likely mate with a wild type. And from there on, what you get is this replacement rate, right? Because a heterozygote, if you guys remember your days of Mendelian genetics, this is going to be 50% het again and 50% wild type. So at best, from that second generation and onwards, the best you can do is replacing. And obviously, you're going to be replacing that very tiny fraction of the mosquito population in the wild, right? So heterozygotes cannot accumulate in a population, and the modified alleles are usually doomed by genetic drift, because for those of you taking 322 now, you know that when the allelic frequency for a given allele is very close to zero, even tiny variations from one generation to another may mean that the allele is lost, right? So all of these experiments of releasing, it's not that people didn't know any better, it's just that we had to try, right? But this idea of going into certain regions, let's say in Africa, and releasing tens of thousands of mosquitoes, they never worked. And here's the reason why. So here's where gene editing and gene drives come in. Wouldn't it be nice if that heterozygote mosquito in the wild somehow became homozygote for that genetic modification that made it resist the malaria parasite, for instance? Wouldn't it be nice if anytime that you have a heterozygote individual, magically it becomes homozygote? Weird, right? Turns out that some people started thinking about these, and this is the idea they came up with, and all of this is experimental yet, but there's been some progress. Generally, you have in a CRISPR construct, you have your sequence for coding Cas9, the enzyme that you need for doing the cutting, your sequence for transcribing that sgRNA that will guide Cas9 to your target site, and then you have your therapeutic or transgene of interest flanked by homology regions that are going to be integrated into your target genome. Right? So after standard CRISPR, what you typically have is one of the homologous chromosomes replaces the target sequence that you cut with CRISPR by these transgene of interest through homologous recombination. What these people, this was work pioneered at UCSD, what these people thought is, wouldn't it be cool if we put everything in between homology regions? Right? So now everything, the rescue sequence that I'm calling there ResX, the guide RNA, and the Cas9 are all under the sequence that will be integrated into the genome. What that does is, after one round of CRISPR that you can do in the lab, you get one of the homologous chromosomes to carry now everything you need to do CRISPR again. So that cell that incorporated one copy of the entire cassette will express the Cas9, express the guide RNA, and express the homologous sequence that you will need to do it again in the other chromosome. About 40 minutes ago, I was telling you how we normally do the opposite. We decouple the ability of retroviruses and of Agrobacterium tumefaciens to do what they do in nature. Here, we went the opposite way. We said, what if we put everything that we need for this gene editing into what is introduced once in a genome, such that once it's been introduced for the first time, it can self-replicate in a way. So you can go from one heterozygote individual to a homozygote individual within a lifetime. And that's how we make homozygote individuals from a heterozygote. Okay? The thinking here is that now the math changes completely. And if you now release 10,000 of these mosquitoes that carry the entire gene drive for resistance to malaria in their genome, they will mate, and instead of producing 100% progeny that is heterozygote, it will be 100% progeny that is homozygote for this thing. Those will mate and produce homozygous progeny again, and before you know it, this thing is literally spreading throughout populations. Hence, obviously, why all of this that we're talking about is still confined to some labs in UCSD, UCI, and some other universities. The feeling of releasing something in the wild that can spread like a wildfire, but genetically speaking, obviously makes many people very nervous. But the concept, at least, is there.